Hi dear students, today we can discuss yeast vectors. Today we can discuss yeast vectors. We can see yeast vectors. The yeast Saccharomyces cerevisiae is one of the most important organism which is used in biotechnology. And the development of cloning vectors for yeast is started after the discovery of 2-muon plasmid that is present in the most strains of Saccharomyces cerevisiae, and only a very limited number of plasmids found in eukaryotic cells. There is only very limited number of plasmid is found in eukaryotic cell. The yeast Saccharomyces cerevisiae is one of the most important organism in biotechnology. And the cloning vector in yeast is developed after the discovery of 2-muon plasmid, which is found in most strains of Saccharomyces cerevisiae. And there is only very limited number of plasmid is found in eukaryotic cells. And now we can see what are the features or what are the genes present in the 2-muon plasmid. In this structure of 2-muon plasmid, this 2-muon plasmid contain gene RUP1 and RUP2 gene. RUP1, RUP2 gene is present in 2-muon plasmid, and that RUP1 and RUP2 gene which is involved in replication of plasmid. RUP1 and RUP2 gene that is present in the 2-muon plasmid that have a role in the replication of plasmid, and another one protein gene that is known as FLP protein. FLP gene is that is present in 2-muon plasmid. And the role of this FLP producing protein is that will convert the A form of this plasmid into B form. That will convert the A form of plasmid into B form. This conversion of A form to B form, that means this FLP producing protein that will rearrange the genes present in the this plasmid. FLP gene coding protein that will convert A form of the plasmid into the B form. That means that will rearrange the genes present in the 2-muon plasmid. And here you can see one gene is the D gene. D gene is there, and the function of this D is not exactly known. This function of this D gene is not depicted yet. And the size of this 2-muon plasmid is 6 kb. Size of this plasmid is 6 kb, and this in each yeast cell that may contain 70 to 200 cells, 70 to 200 copies of this plasmid. In each yeast cell that will contain 70 to 200 copies of this plasmid. And we know that yeast is an eukaryote. Yeast is an eukaryote, and that's why its host chromosome will be coated with the nucleosome. Yeast is an eukaryote, and that's why its chromosome will be coated with nucleosome like that. Like yeast chromosome, this yeast 2-muon plasmid also will be coated with the nucleosomes. Yeast 2-muon plasmid also is coated with the nucleosome. And in the host yeast cell, this replication of this 2-muon plasmid is initiated by the replication enzyme of host cell. And the replication is initiated by the host replication enzyme once per cell cycle. This replication is occur only once per cell cycle. That means replication of this 2-muon plasmid that needs the host replication enzymes once per cell cycle. And it's occur this 2 plasmid having bidirectional DNA replication. Okay, it's having bidirectional replication, and that bidirectional replication is initiated at a specific site in the plasmid that is called as an ARS, that means autonomous replication sequence. Okay, this 2 plasmid is undergo for uh, autonomous replications bidirectional. This 2 plasmid undergo for bidirectional replication, and this bidirectional replication will be initiated as a sequence known as ARS, that means autonomous replication sequence. Okay, we can see again yeast Saccharomyces cerevisiae is the one of the most important organism that is used and that having a wide application in many fields of biotechnology. And the development of cloning vector is in yeast that is started after the discovery of 2 plasmid, which is found in most strains of this yeast cell. And there is only very limited number of plasmids were found in eukaryotic cells. Okay, normally most of the eukaryotes does not contain any plasmid, but only very limited number of plasmids were found in uh, limited number of organisms. Okay, and like that, there is one plasmid, 2 plasmid is one of the plasmid that is present in the yeast. Okay, now we can see the what are the characters of 2 plasmid. Okay, this 2 plasmid that contain uh, gene rep1 and rep2. Okay, rep1 and rep2 that is involved in the replication of this 2 plasmid. Another one protein FLP. This FLP is involved in conversion of A form of this 2 plasmid into B form. That means this FLP coding protein will rearrange the genes present in this 2 plasmid. Okay, and another one gene gene is the that is known as D part is the exact role of this D is not known. Okay, exact proper role of this D part is not known, and this 2 plasmid having a 6 kb in size. Okay, 6 kb in size, and each cell may contain 70 to 200 cells. Okay, each cell may contain 70 to 200 cells, and like yeast chromosome, this 2 circle plasmid will also coated with nucleosomes. I hope you know that nucleosomes. Okay, and the replication of this 2 plasmid is initiated by the host replication enzyme. Okay, host replication enzyme, and there is a in once per cell cycle. Okay, the replication initiated by host replication enzymes once per cell cycle, and in 2 plasmid here you can see this circular DNA. So it is plasmid and covalently closed. That's why okay is occur in the bidirectional. Here you can see here in this like in this 2 plasmid bidirectional replication is occur. Okay, one replication will be go this way and replication will be this way, and this replication is initiated at a site called ARS. Okay, replication is initiated at a site called ARS, that's known as autonomous replication sequence. This is an autonomous replication sequence. And first, in the 2 plasmid based vector construction, we can see what are the markers which is used in 2 plasmid based vectors. Okay, we can see what are the marker genes which is used in 2 plasmid based vectors. Okay, some yeast cloning vectors carry genes which code a resistance to inhibitors such as methotrexate and copper. Okay, some yeast cloning vectors carry genes that give a resistance to inhibitors such as methotrexate and copper. Okay, some yeast cloning vectors that carry genes which give resistance to inhibitors such as methotrexate and copper. But most of the popular yeast vectors that use entirely different type of selection system. Okay, entirely different type of selection system. This most popular yeast vector system does not using resistant genes to inhibitors such as methotrexate and copper as a marker. Instead of that, they are using normal yeast gene as a marker. Okay, normal yeast gene means genes involved in amino acid biosynthesis. Okay, genes involved in amino acid biosynthesis. For example, there is gene LEU2. Okay, LEU2 gene which called for isopropyl malate dehydrogenase. Okay, LEU2 gene that called for isopropyl malate dehydrogenase, one of the enzyme involved in the conversion of pyruvic acid to leucine. Okay, this LEU2 gene is involved in the conversion of pyruvic acid to leucine. Okay, that means this LEU2 gene is having a role in is help for synthesis of leucine. Okay, if you are using this LEU2 gene as a marker, okay, if you are using LEU2 gene as a marker, that time our host cell should be LEU2 leucine oxytroph. Okay, if you are using LEU2 as a marker, that time our host cell should be leucine oxytroph. Okay, or host cell should be leucine oxytroph. Oxytroph means that organism will not synthesize that amino acid. Okay, that organism will not synthesize one amino acid. Okay, that organism will not synthesize a specific amino acid. That type organism is known as an auxotroph. Okay, for that our host cell is a yeast. Okay, our host cell is an yeast, and this host cell should be leucine auxotroph. That's why that means this yeast cell does not contain leucine gene. Okay, this you see here, this is the yeast chromosome, and this yeast chromosome here you can see this is a yeast chromosome. This yeast chromosome does not contain LEU2 gene. Okay, LEU2 gene. That's why this yeast cell cannot survive in the yeast deficient medium. Okay, that means this yeast cell cannot survive media without leucine because this cell cannot synthesize leucine. That's why this auxotroph is a leucine auxotroph. This yeast cell is a leucine auxotroph. Okay, that's why this yeast leucine auxotroph yeast only grow in medium with contain leucine. Okay, only contain medium with leucine. Okay, and when we are when we are transferring this 2-muon plasmid. Okay, when we are transferring 2-muon plasmid, okay, this 2-muon plasmid that contain one gene here, LEU2 gene, that is taken as a marker, and when we are transferring this 2-muon plasmid to this host cell. Okay, this host cell means this is cell is a leucine auxotroph. When we are transferring this 2-muon plasmid to this host cell, that time this host cell will get leucine gene. Okay, leucine LEU2 gene, this host cell will get a leucine gene. That's why this organism is a transformed. Okay, this host cell is a transformed, and this host cell can survive medium without leucine. Okay, that's why this transformed means this cell contain 2-muon plasmid, That's in that chromium you can see LU2 gene, and LU2 gene is will be present in this transformed cell. That's why this transformed yeast cell can grow in medium without leucine. Okay, without leucine it can grow, and this untransformed means yeast cell without this chromium plasmid cannot survive in this medium without leucine. Okay, this is the marker. Okay, that's why LEU2 gene is used as a marker in chromium plasmid. Okay, we can see again some yeast vectors, cloning vectors carry genes that's called for resistance to inhibitors such as methotrexate and copper. But most popular yeast vectors does not use this resistance mechanism genes for the as a marker, but they are using normal yeast or genes, and that's genes involved in the biosynthesis of amino acids. Okay, that's genes involved in the biosynthesis of uh, amino acids. For an example, there is a one gene named LEU2 gene. Okay, LEU2 gene that's responsible for the conversion of pyruvic acid into leucine. Okay, for the conversion of pyruvic acid to leucine. Okay, this LEA2 gene is essential for the conversion of pyruvic acid to leucine. Okay, this one gene that is used as for the as a marker of entomium plasmid based vector system, and this gene is involved in the conversion of pyruvic acid into leucine. That means this gene is essential to synthesize leucine amino acid. Okay, and when we are using leucine as a marker, LEU2 gene as a marker, okay, when we are using LEU2 gene as a marker, that time the host organism, okay, that time the host organism, that means the host yeast cell should be leucine auxotroph. Okay, that time this organism should be leucine auxotrophs. Okay, that means that should not contain functional LEU gene. Okay, here you can see this is a yeast leucine auxotroph. Here in this chromosome, LEU2 gene is not present. That's why this organism should need this leucine auxotroph yeast shall need leucine supplement in the medium. Okay, leucine supplement medium. That means this is cannot survive in the medium that does not contain leucine. Here you can see this is the leucine auxotroph is cell. This cell cannot synthesize leucine amino acid. That's why this this is only survive in the medium that contain leucine. Okay, and when we are using LEU2 gene as a marker, LEU2 gene as a marker, that time will transfer this 2 muon plasmid that contain LEU2 into the leucine auxotroph. Okay, that means this leucine auxotroph is cell will transfer this LEU2, this 2 muon plasmid. Okay, that time this leucine auxotroph is cell will get this functional LEU2 gene. Okay, so this is cell is now is not an yeast auxotroph. Is it can grow in medium without leucine. Okay, medium without leucine. So we can differentiate the transformed cell and untransformed cell by culturing this yeast cell in a yeast deficient medium. So if yeast cell is transformed with this chromium plasmid that contain LEU2 gene, that is can able to survive in the medium without leucine, and if host cell is not contain this 2 plasmid, then then 2 plasmid that cannot able to grow in the leucine deficient medium. Okay, this about markers which is used in yeast 2 plasmid based vectors. Now we can see what are the vectors used in yeast. Okay, what are the vectors used in yeast? First, we can see the yeast episomal plasmid vectors. Okay, yeast episomal plasmid vectors. Okay, vectors derived from 2 plasmids that are called yeast episomal plasmid vectors. Okay, vectors derived from 2 plasmids are called yeast episomal plasmid vectors. Okay, and some YEPs contain the entire 2 plasmids. Okay, some YEPs contain the entire 2 plasmids, and others include just the 2 origin of replication. Okay, some YEPs contain the entire 2 plasmids, and the others include just 2 origin of replication. And example of this YEP is an example of YEP is YEP13. Okay, YEP13 is an example of this yeast episomal plasmid vector. Okay, now we can see the what are the general features of YEP13. Okay, YEP13. First, YEP13 is a shuttle vector. Okay, it's a shuttle vector. It can able to multiply in two hosts. Okay, this shuttle vectors can able to multiply two hosts. That's why uh, it's because it's contain the origin of replication of yeast and E. coli. Okay. It's contain the origin of replication of yeast and E. coli. That's why it's can able to multiply in two hosts, yeast and E. coli. Okay. If any vector can able to multiply in two hosts, then that vector is known as a shuttle vector. Okay. If any vector can able to multiply in more than one host, then that vector is known as shuttle vector. Okay, this YEP13 is a shuttle vector that can able to multiply in yeast and E. coli. Okay, and in the YEP13 vector, the selectable marker is a LUE2 gene is the selectable marker. And this YEP13 also include the entire PBR322 gene sequence, PBR322 plasmid sequence. Okay, the YEP13 also includes the entire PBR322 sequence and can therefore replicate and be selected in both yeast and E. coli. Okay, this YEP13 contain the entire PBR320 sequence. That's why it can able to multiply and can be selected for in the both yeast and E. coli. Okay, it can able to replicate and we can select in both yeast and E. coli. Okay, and then what are the use of shuttle vector? Okay, you can see what are the use of shuttle vector. Shuttle vector can help to recover recombinant DNA from E. coli. Okay, shuttle vector helps to recover recombinant DNA from E. coli. Okay, because it's difficult to recover the recombinant DNA from the transformed yeast colony. Okay, it's difficult to recover recombinant DNA molecule from transformed yeast colony. That's why we can, if you are using shuttle vector of yeast vector, that time we can easily recover recombinant DNA from E. coli. Okay, and we have told that the recovery of recombinant DNA from the each cell is difficult, but in the case of YEPs, okay, in the case of YEPs, the isolation of recombinant DNA from the host cell is not a difficult process because this YEPs exist in a yeast cell as an independent plasmid. But in other yeast vectors, this recombinant DNA will be exist as integrated form in the host chromosome. Okay, integrate the host chromosome, but this YEP will not does not integrate into the host chromosome, that will exist as independent plasmid. That's why recovery of this recombinant YEP from the host cell is not a difficult process. And the word episomal, okay, in the case of YEP, Yeast episomal plasmid, there is a episomal is there. That indicates that this YEP can able to replicate as an independent plasmid. Okay, this YEP can able to replicate as an independent plasmid, but also it's indicates that, okay, this YEP can able to replicate as an independent plasmid, but it also indicates that this YEP can integrate the host chromosome. Okay, this YEP can able to integrate the host chromosome. Okay, that's why it's called as a yeast episomal plasmid. Okay, the YEP can this also indicates that this YEP can also integrate the host chromosome. That's why episomal means the plasmid integrate to the host chromosome. There is episomal. Okay, that means this YAP plasmid can able to replicate as an independent plasmid in the host cell, but it also indicates that this YAP can integrate in the host chromosome. Okay, and this integration is occurs by homologous recombination. Okay, this integration is occurs by homologous recombination between the plasmid LEU2 gene and the yeast mutant LEU2 gene. Okay, here you can see this is the vector LEU2 gene, and this is the host chromosomal mutant LEU2 gene. Okay, this will be mutated one. That's why the host cell will be accepted. Okay, this LEU normal LEU2 and mutant LEU2 will be controlled for homologous recombination. That's in that way this recombinant DNA molecule, recombinant YEP will be integrated into the host chromosome. Here you can see the recombinant DNA molecule integrate the host chromosome. Okay, this is the YEP. Okay, yeast episomal plasmid vector. And the next plasmid, and the next vector is a yeast integrative plasmid. Okay, yeast integrative plasmid is the next vector, that is also known as YIPs. YIPs. Okay, actually this YIPs are basically bacterial plasmid carrying an yeast gene. Okay, YAP gene is almost is contain only bacterial plasmids, and it's also carry a yeast gene. Okay, it also carry a yeast gene. An example of YAP is a YAP5. YAP5 is an example of yeast integrative plasmid, which is a PBR322 plasmid sequence with inserted URA3 gene. Okay, this is a PBR322 plasmid sequence with inserted URA3 gene. Okay, this other is the YAP5. Okay, YAP5 is an example of yeast integrative plasmid that it's contain PBR322 nucleic sequence, plasmid sequence with an inserted URA3 gene. Okay, URA3 genes, actually URA3 gene is present in yeast. Okay, yeast, and this URA3 gene is code for orotidine 5-phosphate decarboxylase. Okay, URA3 gene is code for orotidine 5-phosphate decarboxylase. Okay, with that enzyme is involved in the synthesis of pyrimidine nucleotide. Okay, that enzyme is involved in the synthesis of pyrimidine nucleotides. Okay, this URA3 gene is a URA3 gene code for an enzyme called orotidine 5-phosphate decarboxylase, which that enzyme is involved in pyrimidine nucleotide. Okay, and this URA3 gene is used as a marker. Okay, URA3 gene is marked as a marker in exactly the same way as a LEU2 gene. Okay, LEU2 gene. We already told that LEU2 gene in this YEP plasmids. Okay, LEU2 gene gene involved in the sense of leucine. Okay, leucine. Okay, that means we'll insert foreign DNA, this yeast vector, into the host yeast cell that does not contain LEU2 gene. Like that, in this case, the host cell will be URA3 mutant. Okay, URA3 mutant. Okay, URA3 mutant. That's why this URA3 mutant, in the URA3 mutant host cell, will insert this YIP plasmid. Okay, and this YIP cannot replicate as a plasmid. Okay, this YAP plasmid cannot replicate as a plasmid. It does not contain any part of 2 plasmid. Okay, this YAP cannot replicate as a plasmid, and it does not contain any part of 2 plasmid. And to survive, this YAP plasmid should integrate into the yeast chromosomal DNA. Okay, because this YAP cannot replicate in the host cell as an independent form. For survival, for replication, this YAP should be integrated into the host chromosomal DNA. Okay, if this YAP does not integrate into the host chromosomal DNA, then YAP cannot be replicated. And for replication and survival, this YAP should be integrated to the host chromosome of DNA, then only it can able to replicate. And this integration occur by homologous recombination. Okay, this integration occurs by homologous recombination. Okay, this integration is occurred by homologous recombination. This is about YAPs. Okay, actually this YAP vector is a basically plasmid, bacterial plasmid that carry a yeast gene. Okay, bacterial plasmid that carry a yeast gene. Okay, for example, YAP5 is an example of YAP plasmid, and that contain the sequence of PBR322 and with an inserted URA3. Okay, that YAP5 having PB and a PBR322 plasmid sequence, and it also contain inserted URA3 gene. This URA3 gene is taken from the yeast, and URA3 is used as a selectable marker like LEU2 gene. Okay, URA3 gene also is used as a selectable marker like LEU2 gene. Okay, both the selection mechanism is exactly same. In the LE2 gene, we told that this oxotrophic yeast host chromosome will be used for the recombinant technology, and in the URA3 gene also we'll use URA3 oxotroph. That means URA3 oxotroph, URA3 mutated oxotroph will be used as a host cell. Okay, and this YAP cannot replicate. Okay, this YAP cannot replicate as a plasmid. YAP cannot replicate as a plasmid, and it does not contain any sequence of 2 plasmid. Okay, this YAP cannot replicate as a plasmid, and does not contain any part of 2 plasmid. And for survival and replication, this YAP must integrate into host chromes, then only it can able to replicate. And the integration of this YAP into the host chromosome occur by the homologous recombination. Okay, homologous recombination. This is about YAP. Next vector is a yeast replicative plasmid vector. Okay, yeast replicative plasmid vector, or YRPs. Okay, YRPs. Okay, actually these YRPs are can able to multiply as an independent plasmid because it's carry chromosomal DNA sequence that includes an origin of replication. Okay, this YRP can able to multiply as independent plasmid because they carry chromosomal DNA sequence that include an origin of replication. Okay, and this YRPs contains several genes, including 1 or 2, which can be used as selectable marker. Okay, this YRPs contain several yeast genes, including 1 or 2 of these genes can be used as a selectable marker. Okay, And YRP7 is an example of yeast replicative plasmid vector. Okay, YRP7 is an example of yeast replicative plasmid vector, and it is made up of PBR322 plasmid sequence plus the yeast gene TRP1. Okay, this YRP7 is an example of yeast replicative plasmid, and it is made up of PBR322 plasmid sequence plus the yeast gene TRP1. Okay, and TRP1 is a marker, and this gene is TRP1 gene is involved in tryptophan biosynthesis. Okay, TRP1 gene is involved in tryptophan biosynthesis. Okay, like LEU2 gene and URA3 gene. We can also use the same mechanism is used in the TRP1 selectable mark. Okay, the selection mechanism of URA3, LEU2 and TRP1 is same. Okay, and the yeast DNA fragment present in YRP7 contain both TRP1 and the origin of replication. Okay, the part of DNA of uh, yeast present in YRP7 contains the both TRP1 sequence and origin of replication nucleotide. Okay, now as we can see yeast replicative plasmid. They can able to multiply independent as an independent plasmid in the host cell because they carry chromosomal DNA that contain origin of replication. And this YRP contains several genes, including one or two, which can be used as a selectable marker. And YRP7 is an example of replicative plasmids. It is made up of PBR322 plus the yeast gene TRP1. Okay, plus its YRP7 is an example of yeast replicative plasmid that contain PBR322 plasmid sequence plus TRP1 gene sequence. And this gene which is involved in tryptophan biosynthesis. Okay, this gene is involved in tryptophan biosynthesis used as a marker. Okay, like in YIP and YEP, we have seen LEU2 gene and URA3 gene. But the same selection mechanism used in the case of TRP1, and this TRP1 is involved in biosynthesis of tryptophan. And the yeast DNA fragment present in YRP7 contain TRP1 and the origin of replication. Okay, this is the yeast replicative plasmid. So we have discussed about YEP, YAP, YRP. Okay, we have discussed about YEP, YAP, and YRP. Three vectors, yeast vectors we have discussed. So we have confusion. Which vector is most suitable for? Which is the vector is the most suitable yeast vector? Okay, it is the most suitable yeast vector. So to find out the most suitable yeast vector, there is three factors considered to select the most suitable yeast vector. Okay, to select the most suitable yeast vector, there are three factors is considered to select the most suitable yeast vector. The first of this first factor is transformation frequency. Okay, transformation frequency. Transformation frequency is a measure of the number of transformations that can be obtained per microgram of plasmid DNA. Okay, transformation frequency means number of transformed cells is formed after the transformation process. Okay, in this transformation frequency, YEP is episomal plasmid having the highest frequency, transformation frequency. That means 10,000 to 1 lakh transformer cells will be formed using this YEP. And in the case of YRP, the transformation frequency means 1000 to 10,000, and YIP is less than 1000. Okay, YIP is the less than 1000. Okay, the first factor is the transformation frequency, is a measure of number of transformations that can be obtained per microgram of plasmid. Okay, that means number of cells, transformed cells formed after the transformation process. And the YEP give the highest transformation frequency, 10,000 to 1 lakh cells, and YRP gives 1000 to 10,000, and YIP gives less than 1000 transformations. Okay, and the second factor is the copy number. Okay, which vector having the highest copy number. The YEP and YEP having 20 to 50. Okay, 20 to 50 copies is formed by this YEP, and YRP having 5 to 100 copy, and YAP will be integrated into host chromosome. That's why only one copy will be per cell. Okay, copy in the case of copy number, YAP having 20 to 50, YRP having 5 to 100 copy, and YAP will be yeast integrative plasmid will be integrated to host chromosome. That's why there will be only one copy. Okay, this is the second factor. And the third factor is the stability of recombinants. Okay, stability of recombinants. Okay, this YEP and YRP. YEP and YRP is exist as a independent plasmid. Okay, YEP and YRP is exist as a independent plasmid. But this YIP, yeast integrative plasmids, that will integrate directly into the host chromosome. Okay, that will integrate directly into the host chromosome. That's why that will give more stability. Okay, that's why YIP will give more stability. So in recombinant DNA technology, we prefer which is the vector is give more stable recombinant cell. Okay, that's why we'll select YIP. Okay, because YIP will integrate in the host chromosome, so it gives more stability. Okay, that's why when we are using yeast vectors, YIP is the most suitable vector. Okay, the first when we are considering about the most suitable yeast vector, we will take of three factors. One is the transformation frequency. Transformation frequency means number of transformed cell is formed after the transformation process. In the case of YEP, there will be 10,000 to 1 lakh transformed cells will be present, and in the case of YRP, 10,000 to 10,000 will be this. But in the case of YAP, only less than 1,000 transformation of transformed cells will be formed. And the second factor, copy number. This YEP having 20 to 50 copies per cell, and YRP 50 to 100 copies per cell, and YIP yeast integrative plasmid that will integrate the host chromosome. That's why only one copy will be present in the host cell. Okay, and the third factor is stability of the recombinant. Okay, since this YRP and YEP will be exist as an independent plasmid. YRP and YEP exist as independent plasmid in the host cell, but YIP will be integrated into the host chromosome. YIP will be integrated into host chromosome. That's why YIP is give the most stable recombinants. YIP will give the most stable recombinant. That's why in RDNA technology, there one ever wish to use YIP yeast vector. Okay. Now we can see the yeast artificial chromosome YAC. Okay, yeast artificial chromosome YAC. YAC is one of the important vector of yeast. Okay, and YAC vector present a totally different approach to the gene cloning. Okay, YAC vector present a totally different approach to gene cloning, and routinely it is used to clone around 600 kb DNA. Okay, routinely is it is used to clone around 600 kb of DNA, and the development of YAC is a derivative of the research in structure of eukaryotic chromosome. Okay, the development of YAC is originally derived from the research in the structure of eukaryotic chromosome that work that have identified the key components of a chromosome as being. Okay, actually the development of YAC is derived from the fundamental research in the structure of eukaryotic chromosome. Okay, and that means the structure of YAC is somewhat similar in the eukaryotic chromosome, and YAC contain, okay, this YAC contain URA3 gene, TRP1 gene, sub4 gene as the selective marker. Okay, YAC contain URA3, TRP1, sub4 as a selective marker, and it's contain ARS autonomous replicative sequence. Okay, and selective marker sub4, which is a selective marker in which new DNA inserted during the cloning experiment. Okay, in the sub4 is a selective marker, and sub4 produce red pigment colonies. Okay, sub4 gene helps to produce red pigmented colonies. Okay, sub4 helps to produce red pigment colonies, and during the recombination process, the foreign DNA will be inserted into the sub4 gene. So this transformed colony will not produce red pigment because sub4 will be inactivated. And another one gene is a sen4. Sen4 means which is the DNA from the centromere region of chromosome 4. Okay, this sen4 DNA sequence is taken from the chromosome 4 centromere region. Okay, sen4 is a sen4 DNA is taken from the chromosome 4 of yeast centromere region. Okay, and this sen4 region is required for the chromosome to distribute correctly to the daughter cell during the cell division. Okay, this sen4 helps to distribute daughter chromosome during the cell division. And another one is another gene part is a telomere part. There is another TEL part, which provided by two sequences called TEL, and the structure at the end of the chromosome, and which is needed to the end replication. Okay, which is needed for the end replication of this YAC, and which also prevent the chromosome from being nibbled away by exonucleus. Okay, nibbled by the exonucleus. Okay, this TEL sequence helps to prevent the chromosome from exonucleus, and it also help for end replication. Okay, this YAC vector is a totally different approach in gene cloning. Okay, this YAC vector present totally different approach to gene cloning, and it helps to clone around 600 kb of foreign DNA. And this YAC plasmid YAC vector is developed as a derivative of the research of eukaryotic chromosomal structure. Okay, this YAC vector is formed as a derivative of the study of eukaryotic chromosome structural studies. Okay, and this yeast artificial chromosome YAC contain following selective markers: TRP1 sub4. Okay, URA3 TRP1 sub4. Okay, here you can see sub4, and you can see URA3 and TRP1 you can see. Okay, and this sub4, okay, this sub4 gene that will provide pink, that is produce pink colonies. Okay, sub4 is responsible for production of pink colonies, and at the time of foreign DNA insertion, that time this foreign DNA will insert in the sub4. That's why this sub4 will be mutated, and this colony cannot able to produce red pigment. Okay, this gene cannot able to produce red pigment. Then foreign DNA inserted, sub4 will be inactivated. Okay, and foreign DNA will be inserted in the sub4. And another one portion is a sen4. Okay, sen4 is a part of chromosome centromere of the chromosome 4 is taken from the yeast chromosome 4. Okay, this sen 4 is taken from the yeast chromosome 4. Okay, the sen 4 is taken from the centromere portion of yeast chromosome 4, and this part helps uh, for the distribution of chromosome during the cell division. Okay, this part helps to distribute the chromosome during the process of cell division. And here you can see a TEL. Okay, this is a telomere part, TEL part. This telomere part is essential for the end replication. Okay, telomere part, like eukaryotic chromosome, this telomere part is also helps to for the end replication, and it also helps to prevent the DNA, this yak DNA from the exonucleus. Okay, this telomere part will helps to prevent the uh, this yak from the exonucleus. Okay, these are the structure of yak, and the yak structure is uh, almost similar to the this eukaryotic chromosome. That's why it's called as a yeast artificial chromosome. Okay, yeast artificial chromosome. This is the structure of yak. Now we can see how we'll clone a foreign DNA in the yak. How we'll clone a foreign DNA in yak. For that, first we have to cut this yak DNA with SNAP-B1 and BAM-H1. Okay, first we have to cut this yak DNA with BAM-H1 and SNAP-B1. Okay, we have to treat this yak vector with SNAP-B1 precision endonuclease and BAM-H1. Okay, BAM-H1, SNAP-B1 we have to cut. So that time we'll get 3 fragments. Okay, BAM-H1 will cut here, and here also BAM-H1 cut, and SNAP-B1 will cut here. And that time we'll get 3 fragments. One is up to here, one is this fragment, and second one is this, and third one is this. Okay, third one, and we'll discard this fragment. Okay, we'll discard this fragment, and we'll get 2 fragment like this. Okay, we'll discard and we'll get only this fragment, this and this fragment. Like here you can see 1 and 2. Okay, left arm and right arm we will get, and it's both end will be the telomere. Both end will be telomere, and here there's a SNAP-B1 cut site. Into this SNAP-B1 cut we'll insert a foreign DNA. Okay, we'll insert to SNAP-B1 site. That means here the sub4 sequence is there. Okay, actually this is a sub4 sequence, and we'll cut here in sub4 sequence, and we'll insert a foreign DNA here. Okay, and now we'll get a recombinant DNA. Okay, in the sub4 sequence we insert a foreign DNA, and here 2 and 1 telomere is there, and here also telomere is there, and ura3 is there, TRP1 centromere also is there, and this is a recombinant DNA. We'll insert this recombinant DNA into the double oxotrophic mutant yeast host cell. Okay, we'll transfer this recombinant yak into the double oxotrophic mutant yeast host. Okay, double oxotrophic means that host cell does not contain TRP1 gene and ura3 gene. Okay, that host cell cannot able to synthesize TRP tryptophan and pyrimidine. Okay, that cell cannot able to synthesize TRP1 and ura3. So we transformed this recombinant DNA to the double oxotrophic mutant host. That time when this recombinant DNA is entered in the double oxotrophic mutant yeast cell, that time when is the recombinant DNA is entered, that time the cell will be converted into cell can able to convert tryptophan, synthesize tryptophan and this pyrimidine, so it can able to survive in the minimal medium. So this we'll insert this for the recombinant DNA into host cell, and it can able to survive in the minimal medium. So if this this cell cannot able to survive in the minimal medium, that indicates this vector is not transformed. Okay, and then we have to check whether this foreign DNA inserted into the vector, whether this foreign DNA is inserted in this vector. If the foreign DNA is inserted in the vector, that sub4 will be inactivated. Okay, sub4 means sub4 gene will give red pigment colonies. Okay, red pigment colonies. Okay, if sub4 it is foreign DNA this inserted into the sub4. Okay, foreign DNA is inserted in here, then sub4 will be undergo for inactivation, insertional inactivation, that cannot able to synthesize which colonies, red pigmented colonies. That's why this foreign DNA is inserted sub4, then that colony will be white. Okay, colorless, like blue white screening here. That if foreign DNA is inserted sub4, that time that will only produce colorless colonies. If foreign DNA is inserted into sub4, that means this colony will be pigmented. So we can see, we indicate that foreign DNA is not inserted. Okay, in yeast cloning strategy, first we will cut the this yak with snap B1 and BAM H1, snap B1 and BAM H1. This snap here, that will make one cut, and here also one cut, here also cut. Then we'll get three fragments. BAM H1, here one fragment, here one, and one fragment here. 1, 2, 3. And we'll discard this fragment. Okay, we'll discard this fragment, and we'll get a left arm and right arm. We'll get a left arm and right arm. Okay, and left arm and right arm, and both and will be a telomere, and we'll insert foreign DNA to the here snap B1 cut side. There's a sub4 side. Okay, snap B1 side, there's a sub4 cut side, and here you can see sub4 one fragment is there and sub4 one part is there, and we will insert the foreign DNA, and after that we'll insert this recombinant DNA yak into the double oxotrophic mutant. Okay, double oxotrophic, that means double oxotrophic mutant yeast host cell. Okay, that means that yeast cell cannot able to synthesize tryptophan and ura3 protein, ura3 gene coding protein, that means they cannot able to synthesize pyrimidine. That's why it cannot able to synthesize in the minimal media. For to synthesize this double to survive this double oxotrophic mutant yeast cell, the cell should contain tryptophan and pyrimidine. Okay, and to this double oxotrophic mutant yeast cell will transfer this recombinant yak DNA. So that double oxotrophic mutant yeast cell will be converted into the normal cell, because that will get TRP1 here and ura3 here. That then that cell can, the transformed cell can able to survive in the minimal media. That means media without the tryptophan and the pyrimidine. So it can, so we can easily identify the transformed cell by culturing them in the minimal media. If it is culture, it can be able to grow in the minimal media, that time you can see that that contain the vector. Okay, then we have to identify whether this vector DNA contain the insert. Okay, whether the vector DNA is contain insert. For that we'll check. We told that this foreign DNA insert the sub4 gene. Okay, sub4 the gene. That's why when we insert the foreign DNA sub4 gene, then sub4 gene will be undergo for insertional inactivation. Okay, sub4 gene will be go undergo for insertional inactivation. Okay, sub4 gene will go insertional inactivation. So when the foreign DNA inserted into the sub4, that time this sub4 will be inactivated, so it does not produce red colonies. Okay, red pigment. Red colonies means red pigment does not produce red pigment, so it does not produce red colonies. Okay, if foreign DNA is not inserted into sub4, then what will be happen? That will produce red colonies. Okay, otherwise if the foreign DNA is inserted to sub4, that will produce colorless colonies, like blue white screening, like blue white screenings does not produce colorless colonies. If foreign is inserted to subcrop, then it produce colorless colonies. If not inserted, that will produce colored, red colored colonies. Okay. So this is the yak and the structure of yak and properties of yak and the cloning strategy which is used in yak. Okay. Uh, I hope you understood the yeast vectors and what are the different type of yeast vectors. We explained about YEP, YRP, YIP, and finally we have seen that YAC, okay, yeast artificial chromosomes. Okay, these are the vectors that is related to yeast cloning, where the vectors that are used for yeast cloning. I hope you understood this yeast cloning vectors. Thank you for listening.